Hey guys, it's Mrs. Esser, and today we're going to be talking about our fourth and final macromolecule called nucleic acids. Our nucleic acids are made up of many different types of elements. So here we can see carbon, hydrogen, oxygen, phosphorus, and nitrogen. So this is the macromolecule that's going to have the most elements present. So we remember carbs, CHO, CHO, lipids, CHO, CHO. Proteins, CHON, CHON, and then nucleic acids would be our CHOMPs, so CHONP and sometimes S. So nucleic acids are polymers, and they have these monomers called nucleotides. So nucleotides are going to be easy for us to remember. Nucleic acid, nucleotide, they both kind of have that nuclei part there for you to remember. And our nucleic acids are going to store and transmit hereditary information. Hereditary information is basically the same as saying our genetic information. It's information that's passed down from parent to offspring throughout many different generations. So the three types we'll be focusing on today are nucleic acids, our DNA, which probably we're the most common with, deoxyribonucleic acid, RNA, which we're going to see a lot more of in our protein synthesis unit, and this molecule called ATP that will be used a lot throughout our biology curriculum. So looking at the structure of our nucleotide, every single nucleotide will be made up of three parts. So first thing you're going to look for is this pentose sugar, which we recognize from our carbohydrates unit because any pentagon or hexagon will be a sugar. But this pentose sugar won't be by itself. Instead, it will have a phosphate group attached to it. So something that could help you out would be looking for that P, the phosphate. And then on the other side, you're going to see a nitrogen base. So I can see the N element standing for nitrogen. And depending on what type of nucleotide it is, these nitrogen bases could differ. It's also common where you may see the nucleotide facing the opposite way, where we'd have our pentose sugar, and instead the nitrogen base would be on the left and the phosphate group on the right. But you're always going to see that pentose sugar in the middle. So here's just kind of seeing our monomer versus our polymer of our nucleic acid. So here's one nucleotide, one monomer with my phosphate, sugar, and nitrogen base. And I can link these nucleic acids together to make a chain of nucleotides. So between these phosphate and sugars, it's going to create what we call a backbone. And these will be connected by very strong phosphodiester bonds. So you can think of the backbone as your own backbone. Hopefully you want a strong backbone so that it never breaks. Same thing with our RNA or our DNA. We always want that backbone to be strong. So here's our first example of a nucleotide, ATP, which also stands for adenosine triphosphate. So comparing it to our first nucleic acid, I can see my nitrogen base, in this example would be adenine, my pentose sugar, but here instead of just having that one phosphate group, I have 1, 2, 3, which is where we get that triphosphate in our adenosine triphosphate. So ATP, this is going to be an energy carrier, important for metabolism. We're going to see ATP a lot in our cellular respiration unit. But this is basically energy for our body. So in order to use this energy, our body is going to take this ATP and it's going to break this last bond off. So remember in bio, energy is stored in chemical bonds. When we break those bonds, that's when that energy is used. So then it goes from ATP, adenosine triphosphate, to this phosphate being erased and becoming ADP, adenosine diphosphate. So this will be an important molecule later on that we will talk a lot more about. So just right now associating ATP, which will be an energy molecule, and a nucleotide. Our next one is our DNA, which is huge in biology. We're going to see it in our genetics. We're going to see it in our protein synthesis unit. But to understand our genetics, biologists, chemists, we had to learn this thing called a gene. So a gene is a very small segment of DNA that can turn on and off. So yours and my DNA are very, very similar, but we have certain parts that are coded for and certain parts that aren't. So even though I have green eyes, perhaps I carry the trait for brown eyes, but that gene could be turned off within my DNA. So we'll be looking a lot at DNA in our genetics unit, but DNA stands for deoxyribonucleic acid. Deoxyribose is the type of 5-carbon sugar. And then we know it's a nucleic acid, so that's where we see the Na part of our DNA. So I always think of DNA as the queen bee. One of my uncles owns a beehive, and he's always worried about the queen bee. She needs to be the healthiest. She's kind of the say-all, be-all of the hive. So same thing with our DNA. Our DNA is stuck inside the nucleus. It will never leave. And it's the say-all, be-all. We take our DNA and we take those small segments of DNA, those genes, and they're going to code for other traits in our body. So some characteristics of our DNA. It's a double helix. So basically that's like if I were to take a ladder and just twist it. So I can see the rungs of the ladder, the middle part, are made of our nitrogen bases, which are adenine, thymine, cytosine, and guanine. And then on the sides, we have our sugar-phosphate backbone, which remember, we want our backbone to be very strong. So those are going to be connected by our strong phosphodiester bonds. So just some characteristics of our DNA, our double helix, that twisted ladder. Our sugar phosphates making up the two sides, as I said, connected by the phosphodiester bonds. And on the inside, we're going to have these nitrogen bases. So looking at this picture, you're going to notice A and T are always paired together, so this light blue and this light green. And G and C are always paired together. So these are connected by hydrogen bonds. It was shocking to the chemistry teachers at my school. They say hydrogen bonds aren't real, true bonds. You can think of them more like a magnet. If I put my magnet on my whiteboard, I can take it off again. So same thing with our DNA. Although these bases are connected by a hydrogen bond, we can pull the nitrogen bases apart and put them back together, which will play a huge role in DNA replication later on. So here's just taking a closer look at our different nitrogen bases. We have guanine and adenine, which are purines. I always remember pure and gold, or pure as gold, whatever floats your boat. And then our pyrimidines are cytosine and thymine, which are present in DNA. And then this base uracil we're going to see in our RNA. RNA does not like thymine, so instead uracil will be taking its place. So that rule I listed earlier, saying A always pairs with T and G pairs with C, was created by a scientist whose last name was Chargaff. So we call it Chargaff's base pairing rule. And we can see our A and T pair together, and we have these two weak hydrogen bonds, and G and C pair together, and we have our three weak hydrogen bonds. So just some comparisons between our DNA and RNA just to get you started. We'll be looking at these a lot more later on this semester, but DNA, that double helix, twisted ladder. The nitrogen bases present will be adenine, thymine, guanine, and cytosine. A pairing with T, think of apples in the tree, and cytosine with guanine, cars in the garage. The sugar is a deoxyribose pentose sugar. Remember, anything ending in OSE will be a sugar or a carbohydrate. Location in the nucleus only. The queen bee doesn't leave her hive, and DNA does not leave the nucleus because it's in charge of all the genetic information for our body. Our RNA will be focusing on during our protein synthesis unit. It is single-stranded, so instead of having two backbones, it's kind of like half a ladder. It's going to have one sugar-phosphate backbone. And as I said, that uracil is going to take the place of thymine. So now A will go to U and G will go to C. The sugar is ribose, ending in OSE, so I know it is a sugar, and that's how we get the name ribonucleic acid. The location for the RNA is the nucleus and the cytoplasm, and technically the ribosome, because the RNA will travel from the nucleus through the cytoplasm to the ribosome in order to share genetic information in our protein synthesis unit. So its function is genetic coding of the DNA and creating proteins and protein synthesis. The three types we'll learn during protein synthesis, we have tRNA, which stands for transfer RNA. Its job will be to transfer the amino acids to the ribosome. mRNA, which acts as a messenger for the DNA, which is stuck inside of the nucleus. So you could think of the mRNA as one of those worker bees. And then rRNA, which stands for ribosomal RNA, which helps build our ribosome. So here's just looking at a picture, DNA versus RNA. You can see that single strand versus double strand. Taking a look at the different bases, we have uracil taking the place of thymine. So that's it for our nucleic acids. We'll be diving more deeper into nucleic acids as the semester goes on, but hopefully you learned a lot. See ya.